Hello, I'd like to talk to you guys today about some important information regarding proteins and how they're vital to us in our everyday life. The protein for my paper is over DNA polymers. Proteins, trying to figure out where I should put my face. I'll just focus on this right now. But proteins regulate cell growth, they organize the cell, they have so many different functions, and you can't really just put that all on one slide, everything that proteins do and how many proteins there are, especially how many proteins go into creating DNA. However, there is just this protein, the DNA polymerase protein, that is the one that synthesizes and replicates DNA. It was first discovered, there's a couple different scientific articles that have alternating information between 1956 or 1958. So basically in the late 1950s is when Arthur Kornberg and his team of scientists were able to study E. coli and its effect on, or like DNA synthesis through E. coli, and the DNA polymerase 1 is the one that they were studying. And although more discoveries have been made providing that there are at least 5 different types of DNA polymerase, this is the first one that was discovered. And DNA polymerase 1 and 3 are involved in the replication, while 2, 4, and 5 play more of a repair role of like the damaged DNA. And polymerase 1 is primary structure. So polymerase 1 primary structure is a single peptide, and polymer 2 consists of 7 subunits and aids in the exonuclease activity, which we'll talk about more later, but basically that's where it's repairing the damaged DNA. By studying the replication process, Arthur Kornberg and his colleagues, they were able to isolate that protein, and the DNA's polymerase protein is the ultimate director of cells and is the coder for all living genes through DNA replication. This protein replicates all different types of new copies of DNA genomes and are vital to the development of eukaryotic and prokaryotic cells. They're highly stabilized enzymes, and they're extremely heat resistant. So I'm just going to skip around a little bit. These are the nucleotides that are in DNA synthesizing, and I have adenine and thymine joined together because those are the ones that pair well together. They're complementary to each other in a way, and then the cytidine and the guanosine, well, I can't speak, those pair together as well. And you see errors in DNA synthesis when like adenine combines with cytidine, the C. So when A gets with the C, that's kind of a mistake, and that's where like mutations come from. So basically the requirements for DNA synthesis, you need these 4 things, and without these 4 things, DNA replication cannot occur. And so magnesium ions are vital, and they serve as types of, I think it's like they're a type of primer. And then you need your single strand DNA template, which is the original double helix bond that was broken apart, and then you just need that single strand template so that it can start the DNA polymers can start replicating and multiplying that DNA. And then you need 4 DNTPs. You need all of the DNTPs. There's 4 of them. And And they have like really long names, so I'm not going to go into all different four of the DNTPs, but you basically need all of them. And then you need that primer with the 3OH so that it can combine to it. And so DNA polymerase they resemble more of an open hand. They have they have a thumb, a palm, and fingers. So the fingers are inserted interactions between the nucleotide triphosphates and the existing template base. The palms catalyze the transfer. The palm catalyzes the transfer of phosphoryl groups and reactions and catalyzes the essential amino acids in the active sites, while the thumb assists in aligning the double-stranded DNA. And it looks something like this. Genes are like the story, and DNA is the language that the story is written in. I went through this slide already. The replication fork is basically whenever the two whenever the DNA, the helix is being separated, it's kind of like going into a fork shape, and the leading strand is there at the bottom, and that's from the 5' to the 3', and that's the only way that DNA polymerase can be in a continuous motion where they just go in one big long segment, while the lagging strand featured up top is the 3' to the 5', and that is where the DNA polymerase is working in not a continuous, it's a discontinuous way. And the reason why it's discontinuous is because it's trying to find in my notes here, but basically I'm trying to read my paper, but basically the reason why it is discontinuous is because it's making these small fragmented sections, and it's having to go back and pair them and double check that everything's correct. It's a lot slower in this lagging stage, and it's also where more mistakes are going to be made. Basically the RNA primers, they have to be joined to the opening of the replication fork, and it's exposed to the new template. Basically, this produces a series of disconnected, they're called Okazaki fragments, and the Okazaki fragments are, during mutation, it's used to prevent the possibility of deleterious activity among the peptide factor, and this is basically saying that it's trying to prevent mutations from occurring. And basically many barriers must be overcome during the DNA replication because those structures are just so intensely complex, and so they're super small as well. Luckily, the DNA polymerase are able to have they have kind of a proofreading to them, and that's where it's the polymers 2, 4, and 5 come into play, and they're able to check to see if there's anything wrong or they've created a mistake. And that's where the polymers 2, the exonucleus activity, really comes into play. And that activity works by moving one step back, and it recognizes the mistake, and it removes that mismatched nucleotide, like I was saying, with the A and C versus it being the A and T. So it recognizes that there's a mistake. It actually detaches that nucleotide and then replaces it with the correct one. However, sometimes those mistakes are not always caught, and in humans, that's actually where cancer comes from. But sometimes that's one of the things that it can happen. But once if it just keeps going and it doesn't, there's no going back. So it can either make the correct fix the mistake, or there's no going back. So it fixes a mistake, or it doesn't, and it starts to make another one. Well, say you're like 2-3 fragments away. Well, now it's way too late. It doesn't remove 3 parts. It can just remove that last protein that was added. There's replication checkpoints in place to prevent such breakages, and those stalled forks, they can resume replication once the obstruction is like relieved. But like I was saying, like if it fails to incorrectly pair those nucleotides, then the mutation becomes permanent. And it no longer views it as an error. Let's see. Oh, and here's just the Ozaki fragments like I was talking about. On the bottom, like, structure, the very, very bottom, it's where it says continuous synthesizing. That's showing you that it's going from, like, the direction that it's heading versus the continuous up top. These are those mutations I was mentioning about. We've got the C right here, and it's, they're trying to pair it up with the T, but that's not where it's supposed to go. The T is supposed to be with the A. And in summary, I forgot to take that slide out, but basically, in summary, there's my literature cited, and then in conclusion, proteins play a vital role in the development of all living organisms, whether they're cleaning up, sorting, or organizing the intel. And the DNA polymers, being one of those proteins, they synthesize properties that are most critical in the faithful development of DNA. And the DNA polymerase protein is the ultimate director of cells and the coder for all living genes through DNA replication. And thank you for watching. As usual, signing off.